Welcome to the Life Science Success Podcast. For those of you who don't know me, my name is Don, and I'm a consultant in life sciences, and I help companies overall with process improvement and implementing new technologies, as well as things like project management and things like that inside of life sciences. Today, actually, we have a very exciting guest. My guest is Matt Hill. Matt is the founder and CEO of Elegen Corporation, privately held company in the San Francisco Bay Area, committed to revolutionizing synthetic biology workflows with innovative DNA writing technology. And so with that, welcome, Matt.
Thanks so much, Don. Really pleasure to be here.
Yeah, thanks a lot for being here. So if you wouldn't mind, would you just take a moment and tell the listeners a little bit about yourself?
Sure. So currently CEO and founder of Elegen, company that I've been involved with, started back in 2017 and have been leading ever since. I guess my sort of short history here is I have a Ph.D. in genetics, did that at Stanford here in California. Actually came out to the Bay Area originally and Stanford in particular because I was very interested in startups even before going to graduate school. I thought it was a very good ecosystem to put myself into while I pursued that graduate degree. And then after my PhD was finished, I actually went into the startup world. I joined a fledgling company at the time called Natera. Natera is currently one of the leading molecular diagnostics companies. And so I joined that company back in around 2009. It was a fledgling company that had one initial product in the market, and I joined a very small biology team to help them build out new products and new capabilities. Go ahead, yeah.
And then I was going to ask, so then what made you want to go from that to, more the R&D, sort of the development of the product side over to now being a CEO and founder?
Yeah, I am. I guess I'm a consummate builder. I really, or maker, perhaps. I love sort of connecting dots across different technology areas. I love trying to find, I'm that guy who's sort of, if you complain to me about a problem, I'm the guy who tries to immediately start troubleshooting it for you. I tend to bring that to my own work pretty frequently. And so, as I was leading the R&D team over at Natera, there's just a handful of different areas that I got really intrigued about and could see potential new solutions to those problems. One of those was actually in the production of DNA. And it hit us in a couple of different fronts at Natera. One of the key technology platforms that I helped create with my team over there was a massively multiplex PCR technology. And that's effectively a 20,000 plex PCR reaction in a single tube that allows Natera to collect exquisitely rich and precise data about what's happening. At least the initial product there was non-invasive prenatal testing. So rich, very, very rich, very precise data on what's the sort of the genetic state of an unborn child. It was a very exciting opportunity to sort of build out a new technology platform while there. But one of the things that I encountered was, just getting DNA to actually allow us to iterate and experiment on that technology platform was one of the important bottlenecks in our process. We were ordering very, very large collections of oligos in order to do a very large multiplex primer approach. And there was a pretty substantial latency in those. So it was, maybe one of the first times I recognized that DNA synthesis and DNA production itself is still a major bottleneck, a major hurdle in this ecosystem. And it was slowing us down. And when you take a step back and look at what's going across or going on in this entire ecosystem, it's slowing down every life science company on almost every project they're doing. DNA is such a foundational input into every product that gets developed, whether you're talking about therapeutics or ag bio or syn bio. DNA is the starting point for many of those projects. And that's a problem that just needed to be solved. The technologies, for the audience, if they're not familiar, the technologies that are involved in the production of DNA are fairly complex, and many of those technologies are decades old. It turns out to be a multi-step manufacturing process. Most people think of it as DNA, quote unquote DNA synthesis, but the actual production of DNA for or longer double-stranded DNA for rewriting genomes, or re-engineering cells, reprogramming cells is actually this double-stranded DNA, which has multiple steps involved in its production. And I'm happy to get into that at the right point.
Yeah, that'd be great. Where my mind went to is that, I mean, diagnostic companies, I would imagine like Natera, over a period of time, build just an immense, it seems like tissue bank for the things that they, continue to examine throughout time. And so, yeah, I definitely am interested in hearing how this plays a role for companies that are developing diagnostics, because I have seen it myself. A client will come in and they'll say, we would like, to develop a companion diagnostic for a client that has this type of structure, this type of patient, structure. And then you're off, searching for those types of, oftentimes, tissue samples and other things to try and, make sure that you accurately target the diagnostic for the thing that you're looking for. And so, yeah, I've personally seen how it can slow down diagnostic companies in our space as well.
Yeah, indeed. Yeah, that DNA is it's so fundamental to everything that we do. And as I said, the tech employed for creating that DNA is decades old. Especially because there are multiple steps involved. There's been, of course, there's been some innovation on different aspects of that process. There's, the three fundamental pillars for you and the audience. If you're making longer double-stranded DNA, I think everybody's sort of intuitively familiar. If you do biology at all, you're intuitively familiar with primers and probes and oligos and that sort of thing. That's actually the starting point, but it's not the ending, the end point for making double-stranded longer DNA that you might use to reprogram a cell. So you make oligos first, which is the true synthetic step. That's nucleotides to oligos. But then if you want to make longer double-stranded DNA, you actually have to take those oligos and you have to assemble them together into longer double-stranded fragments. And you leverage the hybridization of oligos, of overlapping oligos to achieve that. And then there's a third step in that process, which is purification. We, in the field, know it as cloning. But cloning is really just a purification procedure. When you do that, that oligos synthesis step followed by the assembly step, you inevitably end up with errors, molecules that contain errors. Some of the molecules in that tube might be perfect, but a large fraction of them actually contain errors. And what we all call traditional cloning is a means of basically partitioning those molecules into cells. And each cell, each transformation event picked up by E. coli. So you put a fragment into an E. coli vector backbone, plasmid backbone, and then you electroporate it into an E. coli cell. You put those onto an agar plate with an antibiotic resistance marker, and that allows you to identify single instances or single molecular species. And you leverage the cell, the E. coli cell, of course, itself, to replicate the DNA to produce enough material that you can take it downstream and test and maybe do some sequencing or run some gels. But you've got these three fundamental steps of synthesis, assembly, and then purification. And the cloning step has remained unchanged. I mean, strictly speaking, unchanged since it was invented in the early 1970s. So we're really talking about technology that is many decades old. There are challenges at the assembly step that limit the types and complexity of DNA that you can create. And then there's also some pretty fundamental challenges at the oligos synthesis step that relate in many ways to the throughput and the cost of making such a wide variety of those oligos to feed downstream gene production. So you've really got three fundamental problems that you need to solve, and that sort of notion was the starting point and the origin for Elegen.
Very interesting. So at Elegen, you have a product called Infinia DNA. Can you tell us a little bit about how this technology is revolutionizing synthetic biology workflows?
Yeah, our vision with Infinia was to allow customers, and, researchers and scientists in the life sciences to uh, streamline their workflows. I mean, I think there was a mantra over the last handful of years around, engineering biology with rapid design, build, test, learn, right? Um, you want to be able to execute that flywheel as fast as possible, because we don't necessarily, biology's got a lot of unknowns. We don't necessarily know the operating characteristics of a system that we're trying to engineer. You've got to take a lot of shots on goal rather, and you want to do that as quickly as possible so you can learn with each iterative cycle. It's the rapid prototyping notion that we have in many other fields.
Right.
Engineering has been using this for many, many decades, of course, and we're trying to bring that to biology. And you've got to be able to actually rapidly make the DNA, which is the software for a cell. You've got to be able to make that DNA, reprogram and get it into a cell, reprogram it quickly, take your measurements, and then in many cases, iterate very, very quickly. And so that capability just didn't exist. And so our vision with Infinia DNA is that we streamline that process. We make DNA up to 7,000 bases. Today, we can deliver that in as little as 6 business days. And we can do that with greater complexity and effectively clonal accuracy. And we can get that into customers' hands very, very quickly and allow them to do more faster. So do more interesting science, gain more insights, and then turn the crank and iterate faster to build better products.
So could you give us some real world examples of the applications of the products?
Uh, yeah, I have to be careful not to share customer
Sure.
information, but um, we've got a number of customers in the therapeutics realm exploring different sequences. We've got quite a few customers across the mRNA therapeutics world, for example, who are testing out a variety of different targets, different promoters, different terminators, trying to optimize their expression of their targets. They're iterating through a number of different molecules to optimize their downstream therapeutic benefit.
And then as you look across the different industries that you touch, is there one primary industry that you currently work in, or is it across everything that we've talked about before? Because we had talked about sort of the like agri side, healthcare and pharmaceuticals. So just curious, if there's one industry that really stands out, and then also kind of how they're ranked.
Well, we think there's impact across of our product and both our current and our future product offerings across the entire ecosystem. That is, therapeutics and ag and synbio. We have customers across all three of those broad areas. One of the areas of greatest initial uptake, is in fact in the therapeutics realm. That's probably somewhat driven by the fact that customers are perhaps most eager to move very, very quickly. Time is money, and when you're talking about a therapeutic, time it potentially equals a lot of money. And the ability to get the long, clonal or perfect fragments or strands of DNA that you need to execute your analytics is absolutely essential to some of those programs. So the ability for them to move fast is critical, and so we're seeing a lot of very rapid uptake in that area.
And then, in what ways does your technology contribute to the advancement of the bioeconomy as well?
Oh, I like to think it's foundational. I mean, this is we are an accelerant in that flywheel, and we accelerate the rate at which new companies, existing companies can discover new products, develop new products. And for me, this is the most exciting part of it, is that we view ourselves as really enabling to this whole ecosystem. And the faster they can develop products that actually work. It's a very interesting thing, actually. In the biological sciences, the typical development cycle for a lot of products is 6 to 9 months. You typically have about that long you've got some corporate mandate, executive, senior executive mandate to do something, to solve some problem. And the way it historically works, you perhaps have a couple of ideas of how to address a particular problem at the biological level. And then you'll try and instantiate those and make it work. And you've got really 1, 2, maybe 3 shots on goal to make progress, to show definitive results. We call it feasibility, where I come from. Did your, if you're developing a diagnostic, you want to get to a point where you can show that your particular approach actually yields results that suggest a particular diagnostic approach might work. You've got 6 to 9 months to show that before decisions are made about whether to continue or stop a project. What we want to do is enable the world to see 6, 7, 8 shots on goal in that same time period, and being able to supply the DNA that lets you take those shots on goal instead of waiting 6, 8, sometimes 12 weeks, especially if the DNA is long or complex. Instead of waiting that kind of time frame to then finally get your DNA into a cell and take your measurements and get a result and evaluate the result, we want the entire ecosystem being able to do this on the order of a week or 2, as opposed to a couple of months. And I think that totally and completely shifts the paradigm of the types of products and types of solutions we can create in the life sciences. I mean, to me, this really underpins this entire world, this entire future of biological engineering or programmable biology. It is the thing that a lot of the key opinion leaders started talking about in the early 2000s. We just didn't have the technology to get us there.
Right. Yeah. And it's interesting because you'd love for in a lot of these cases for the, drug discovery to be accelerated. You'd like for the, kind of final product development to be accelerated, but oftentimes we're so challenged with, the availability of information to be able to confirm, the thing that we've developed, whether or not if it's ready, to move to that next step. And it sounds to me like this will be a big accelerant for that in the future as well.
Yeah. And look, and it's not just speed, but it's also scalability and to some extent cost. Our technology allows groups to cast a very wide net right up front, take a lot of different measurements, look at a lot of different options for the sequence, call it promoters, terminators, a variety, variant libraries. These are all things we can do and allow groups to cast a very wide net, take that data, model it, understand it. Obviously, there's a very exciting intersection now emerging around bio and AI, which is really starting to take off. And I think the ability to look at and collect a lot of data is going to be the driving force behind AI, because you can't build AI models without the data in the first place. And then you want to be able to generate more data that feeds back into your AI models, that you can improve those models and get better and better with each turn.
Well, interestingly enough, as well, the timing of our interview coincides with a big press release that you just had today earlier. So would you mind just sharing with the listeners a little bit about what's happening right now with Elegen?
Absolutely, and thanks for giving me the opportunity to share this.
Sure.
So we're excited to announce that here at Elegen that we just closed a partnership, an agreement with GSK. We're very excited to announce this. It is a $35 million opportunity for us, and it supports us in a number of ways, with upfronts, equity investment, and then milestones upon achieving certain technical objectives that they're interested in.
So yeah, it's got to be just a tremendous day for the entire company as well, to just see that all of the hard work that you put in, sort of resulted in this milestone as well.
Absolutely, and I'm very proud of the team. The team's been working exceptionally hard across the board to get us where we are in terms of the state of the technology. And then, of course, it's incredibly validating to have this recognition from a partner like GSK, and validates everything we've built and validates the vector that we're on as a company.
So my last few questions really are going to focus more on you overall as an individual and as a leader. The first one is, what's the greatest leadership advice that you've ever received, and how has it really impacted the approach that you're taking as a founder and CEO?
That's a very good question. I don't know if I have a specific piece of advice, but I've learned by watching, both at my previous company, Natera, really amazing team there. Really, really just tremendous team, brilliant folks. And I think, the ecosystem of innovation that team created is something that still inspires me today. And I've tried to replicate that here at Elegen. I think the idea is, we do things. I'm inspired personally by doing things with impact,
Mm.
and touching human lives, and really wanting to help people. And then, of course, folks like myself, and I'm sure many of your listeners, we went into science because that's also our potential, our technical strengths, our abilities, and so it's this overlap of it's what we like to do, it's what we're good at, and it has a lot of impact. And so I've tried to bring that to Elegen. I try to inspire and encourage my team and foster that at our company. And I try to bring in people who believe and live that sort of ethos every day. And I think that has sort of a very virtuistic flywheel in that I think everybody really enjoys their work and they like working on the very important problems that we are working on.
Yeah, absolutely. And it's interesting because whenever you sort of look from the outside perspective, I feel like there are people that sort of think about the profits and things like that companies generate and sort of, spin this into, oh, well, that's why they got into this. But whenever you talk to so many people, it's no, we started with the patients, and, we're very focused on improving lives. And, yes, there are profitable companies in life sciences, but I very seldom will run into somebody that starts out with hey, I got into this because I was thinking I was getting into the oil industry or the energy industry or, some other industry where I knew I would make a lot of money. Instead, people, I feel like in our space very much do want to transform, the lives with the things that they're bringing to market. And so thank you for sharing that.
Yeah, I think that's absolutely true. And I think, many of us could have, in this field, could have gone on to do things, in the energy sector or the finance sector. And we elected to do the things that we do because we love it, because we were inspired by it, and we love the potential impact that it has on the world around us, and in many ways, a very positive way. And I think, um, of course, any company has to be profit seeking. There's no doubt. You can't circumvent that. But I view the profit seeking as a barometer that you're working on an important problem, because important problems have value when you have a good solution. And so that profit seeking becomes a barometer for what you should be working on. And there's no misalignment. We get to make money for our investors, and we get to come to work every day and and make a real impact on the world.
Such an important point. Thank you for sharing. Next question is, what inspires you?
Um, I think in many ways, I hate to refer to my last answer, but again, I think it is this notion of having a real impact in the world and being able to do things that change people's lives. And so I always, maybe to just continue the thought at least, is I always look for, I enjoy solving hard problems, but I'm always looking for problems that I think do have a very specific impact that you can point to. And, when we talk about Infinia DNA and the technologies that Elegen has developed, these are our customers are in the therapeutic realm, and these pharma companies like GSK and others are developing treatments and therapies that directly impact people's lives.
Mhm.
Our technology could feed into the next COVID vaccine, for example, or personalized therapeutics, which are going to be addressing some of the long-standing biggest challenges that we face, like cancer.
Right.
And to know that Elegen's technology could be absolutely enabling to that solution, to a category of therapies that allow us to really improve patient lives who have cancer or have had cancer. I mean, you've seen, I'm sure your audience, many of the members have seen the recent Moderna data. I mean, that's very inspiring in terms of the success that they're seeing with their combined oncology therapy in
Hmm.
melanoma. So we're very excited to see that develop, and we would, of course, as a company, like to see and become part of that solution as well.
Very good. What concerns you?
Um, I actually am quite frustrated personally by the gap in funding for deep innovation in the life sciences. So the concern is that we're just not doing this at the rate and the depth that we need to support ongoing foundational innovation in the life sciences. And what I mean by that specifically is there's a pretty sizable gap uh, out there in the ecosystem around getting enough capital in to develop novel technical solutions, novel biological solutions to foundational problems. Um, for example, the SBIR world, the grant world, those grants, many of your listeners may experience, they're very hard to get. And the amount of money actually and capital available in those grants is typically pretty small. And the biological sciences is a very expensive field to operate in, and so that money doesn't take you very far. And then, of course, the venture community wants solutions that work, demonstrably work, and so there exists a gap in funding foundational research and development that I don't that we need to actually work to close. So I'm concerned that we're not doing enough. There needs to be more grant opportunities. The grants need to be much larger. Um, I think it's a real problem, and we've got to figure out how to solve it.
Yeah, I don't disagree at all. I mean, yesterday, last year especially, I mean, we heard routinely from companies that had, great products, great results, were at a pivot point for the overall organization, and either they couldn't continue and they had to shelve things until they could, maybe come back at some point this year, or there were certainly companies that just didn't survive. If you think of mainstream companies that are researching, things like cancer drugs and things like that, where those companies seem to have an easier path, um, to find a solution. But if you're a company that develops I don't know, some form of treatment for an eye disease or a CNF disease, those things just are not as well funded. I feel like in the same camp that you are, that one, at some point any one of us could need any of these things, and yet if the very few dollars are only going to one area, it really makes it tough for those other things to try and survive. And so I know that the entrepreneurs are trying to be extremely creative, and I've heard of a lot of great solutions for to at least attempt to try and find funding, but it is a very tough environment.
It is indeed. It is indeed.
Okay, last question: what excites you?
Um, I think coming back to the AI part of this story, as again, as Elegen deploys this capability that allows you to turn that flywheel very quickly, I think the emergence of AI and that intersection between AI and bio is going to result in a world that I think is somewhat unrecognizable in a very short period of time. I mean, this is really an opportunity to, you know, build and design things at the biological level that I don't think we've ever had any notion how to do. And AI is going to drive a lot of that. And of course, Elegen's here to make the DNA that's going to be required to actually reprogram the cells themselves. So you might have models and predictions generated by an AI model, but then you actually have to go and construct it, and that's where we come in. So I think it's going to be mutually synergistic.
I do look forward to um, hearing the scientist in the future sort of criticize the output of the model, and in both ways, right? One, sort of the, hey, look, this helped us, see a little clearer path that maybe we didn't see. But also if you look at what the output of ChatGPT as an example, if I asked it to write an article, for me, I may criticize certain elements of that and think that it should be done differently. Whenever it comes down to the base science and the decisions that we're making, it'll be interesting to see if there's a logical sort of disconnect that maybe would have led us in a different direction, with the solution, and whether that eventually matches, kind of where the endpoint needed to be, or if there's still a disconnect at the endpoint as well. I'm hearing, a lot of great things in terms of in silico research that's ongoing, and certainly, a lot of benefits to AI. I really sort of question that future, and what the output of the scientists and the developers will be whenever they go back and look at the models.
I mean, I see what you're saying. I think the powerful thing about these AI models are that they're frankly just, I mean, they're smarter than us. They can, if you feed them the right data, they can traverse that data and make connections in that data that I think is very hard for humans to do. And while the data isn't quite there for call it the biological sciences, a lot of scientists are going to be pursuing this very quickly, and we're going to start populating that space. I think, just imagine a chat GPT built on top of all existing available journal literature, which
Right.
was not incorporated into the chat GPT, the original chat GPT models. And then imagine when you have some biological question, you've got a supercomputer that can synthesize across all of that journal literature rather than you reading dozens and dozens and dozens of articles, review articles, and primary articles, and trying to synthesize that all yourself. You've got this, this AI system that can. You've still got to prompt it. You've still got to ask the right questions, but then it becomes an iterative process between a Q&A process that advances you through your thinking, and fingers crossed you're getting reasonably accurate responses. And to the extent that you're not getting reasonable accurate responses over the next few years, people will be adding more data to these systems and rebuilding the model, and you've got this very quickly ratcheting improvement in the models. So I'm excited to see what comes out of this.
Yeah, I am too. I am as well. Well, Matthew Hill, thank you so much for joining the Life Science Success Podcast, and thank you for telling us about Elegen and the great work that's going on there. Congratulations, by the way, to you and the team as well on your recent relationship, and I wish you success in the future.
Thank you so much, Don. Really appreciate it, and we all thank you here at Elegen.
Thanks a lot.